The topic of the lecture is the circulatory system. The circulatory system includes the cardiovascular system and the lymphatic vascular system. The cardiovascular system includes the heart and the arterial, capillary, and venous systems. The circulatory system is divided into the macrovasculature. This part contains vessels with more than 0.1 millimeters in diameter, for example, large arterioles, muscular and elastic arteries, and muscular veins. The next, microvasculature with arterioles, capillaries, and postcapillary venules. The circulatory system plays a role in the movement of all body fluids. Circulatory system transports oxygen and nutrients to the tissues. Circulatory system transports carbon dioxide and metabolic waste products from the tissue. Circulatory system is involved in temperature regulation and the distribution of hormones, vitamins, and cells of the immune system. The vessels constituting the cardiovascular system are arteries, veins, and capillaries. The walls of arteries and veins are composed of 3 layers called tunics. The 3 layers of vascular wall from the lumen outward are the following: tunica intima, tunica media, and tunica adventitia. The arterial system contains large, medium, and small arteries and arterioles. This system conducts blood from the ventricles to the capillary networks under higher pressure than veins. The venous system contains large, medium, small veins and venules. Veins collect blood from capillaries and transport it to the heart under lower pressure than arteries. Capillaries are the smallest vessels with the thinnest walls, are interposed between the arterial and venous systems. Capillaries permit the exchange of materials between cells and the bloodstream. Capillaries receive blood from the smallest arteries, the arterioles, and deliver blood to the smallest veins and the venules. Capillaries are the smallest diameter blood vessels, often smaller than the diameter of erythrocyte. They measure from 5 to 10 micrometers in diameter. The wall consists of a single layer of squamous epithelial cells called endothelium, basal lamina, and pericytes. Pericytes are contractile cells associated with capillaries and small venules. They surround the capillary with branching cytoplasmic processes and are enclosed by a basal lamina. Pericytes may act to regulate blood flow, may also function as regenerative cells that play a role in repairing damaged vessels. This is microscopic slide of capillary in longitudinal section. We used here chematoxylin and eosin staining. Arrow A shows pericytes located outside the capillary. Arrow B shows squamous dark purple flattened nuclei of endothelial cells, and arrow C shows erythrocytes which are present inside the capillary. This slide shows a microscopic slide staining with hematoxylin and eosin, and we can see here capillaries and precapillary. The two black arrows show precapillary. The wall of precapillary contains endothelium and smooth muscle cells which are circularly arranged. The nuclei of endothelial cells are longitudinal to the blood flow. The smooth muscle cells are circularly arranged, and because of that, their nuclei are perpendicular to the blood flow. When the nuclei of endothelial cells and smooth muscle cells meet, we have very characteristic histological feature because nuclei of smooth muscle cells are perpendicular to the nuclei of endothelial cells, and it is very easy to recognize in microscopic slide. Histological structure of capillaries varies in different tissues and different organs, and because of that, on the basis of the morphology of capillaries, they are divided into three types: continuous, fenestrated, and discontinuous capillaries. Capillaries contain two types of cell: endothelial cell and pericytes. Endothelial cells are of mesenchymal origin. They are flattened, elongated, or polygonally shaped. They are aligned with their long axis in the direction of the blood flow. They are squamous, very thin cells, but they are a little thicker when the nucleus is located. The nucleus causes the cell to bulge into the lumen of capillaries. The cytoplasm contains typical cellular organelles, small Golgi complex, mitochondria, free ribosomes, and a few rough endoplasmic reticulum. The cytoplasm contains numerous pinocytotic vesicles. Endothelial cells participate in the structural and functional integrity of the vascular wall. The second type, pericytes, are found around capillaries and venules, and they are undifferentiated mesenchymal stem cells. Pericytes share the capillaries basal lamina, form gap junctions with the endothelial cells. The cell body of pericyte forms branching numerous cytoplasmic processes that surround the capillary. Pericytes may regulate blood flow and function as regenerative cells. This slide shows cross-section of capillary, which is lined by endothelial cells. Very thin squamous flattened cells with typical cellular organelles, and the part where the nucleus is located is thicker and bulged into the lumen of capillary. This is electron micrograph with longitudinal section of capillary. We can see pericytes located outside the capillary and numerous branching cytoplasmic processes that surround the capillary. There are three types of capillaries: continuous capillaries, fenestrated capillaries, and sinusoidal capillaries. This classification is based on the morphology of the capillaries. These structural variations in capillaries allow for different types of metabolic exchange between blood and the surrounding tissues. Continuous capillaries are typically present in muscle tissue, in connective tissue, skin, lungs, and central nervous system. They are characterized by an uninterrupted vascular endothelium, endothelial cells resting on a continuous basal lamina. They contain the usual organelles, short microvilli on their laminal surfaces, numerous pinocytotic vesicles, and variable number of electron-dense membrane-bound vesicles. We have here schematic diagram of continuous capillary with uninterrupted vascular endothelium with continuous basal lamina. The endothelial cells are joined by tight junctions that restrict passage of molecules from the lumen into underlying tissue. Fenestrated capillaries are typically present in endocrine glands, kidney, or intestinal tract. The endothelial cells are characterized by the presence of numerous circular openings called fenestrations. The continuous basal lamina is located across the fenestrations on the basal plasma membrane surfaces. Fenestrations in the cytoplasm of endothelial cells designed for rapid exchange of molecules between blood and surrounding tissues. This slide shows fenestrated capillary. Fenestrated capillaries have endothelial cells with numerous fenestrations and they resting on continuous basal lamina. In some organs, fenestrations may have a thin, non-membranous diaphragm across their openings. Sinusoidal capillaries or discontinuous capillaries or sinusoids have discontinuous or missing basal lamina and incomplete endothelial cells perforated by large pores. This type of capillaries are present in the liver, spleen, and bone marrow. They are larger and more irregularly shaped than other types of capillaries. The endothelial cells in the sinusoidal capillaries have large openings in their cytoplasm, and these cells are separated by wide, irregular intercellular gaps. These intercellular gaps allow for passage of blood plasma proteins. The endothelial cells resting on a discontinuous basal lamina. This basal lamina in some organs is rudimental or may be absent. Traditionally, arteries are classified into three types depending on the size and the characteristic of the tunica media. Blood vessels, including all arteries, are composed of three concentric layers. They are the tunica intima. This part contains endothelium and subendothelial connective tissue. The tunica media, usually this part is the thickest of the three layers and is composed of elastic fibers or circularly arranged smooth muscle cells. The tunica adventitia, the outermost layer, is composed of fibroelastic connective tissue. In larger vessels, the tunica adventitia houses vasa vasorum. They are small blood vessels that supply the tunica adventitia and tunica media of that vessel. This slide shows two types of blood vessels: the artery, right, and the vein, left. Please pay attention to the lumen of these vessels. The lumen of artery is regular and open. The lumen of the vein is irregular. In the vein, the layers are poorly demarcated. In the artery, the layers which are present in the wall are very well distinguished and the boundaries are very well visible. The layers in the vein and the boundaries are hardly visible. Tunica media is the best developed in the artery. Tunica adventitia is the best developed in the vein. The veins may contain the valves in the lumen which are absent in the arteries. Arteries conduct blood away from the heart, and they are classified into 3 categories or 3 types. Elastic arteries, also known as conducting or large arteries. Muscular, also known as distributing or medium arteries. Next type, they are arterioles. And last one, they are metarterioles. Large arteries are called also elastic arteries. They conduct blood from the heart to the systemic and pulmonary circulations. They include pulmonary arteries and the aorta, for example. They contain a large quantity of elastic material in their walls. Thick tunica media contains numerous elastic membranes. The internal and external elastic laminae are difficult to distinguish from nearby elastic membranes. The elastic materials enable the vessels to recoil to accommodate pressure changes and maintain a continuous flow of blood during ventricular diastole. We can see here longitudinal section of the carotid artery. This is microscopic slide. The carotid artery has the histological characteristic of elastic arteries. Thick tunica media contains multiple layers of elastic lamina that are organized in concentric fashion. These elastic laminae are sheets of fenestrated elastic material that is produced by smooth muscle cells interspersed between the elastic laminae. The elastic laminae are not well visible with this type of stain. In this specimen, we can see nuclei of smooth muscle cells. The tunica adventitia contains loosely arranged connective tissue fibers. They are collagen fibers, elastic fibers, which are produced by fibroblasts. The tunica intima of the elastic artery is composed of endothelium, subendothelial layer, and internal elastic membrane. The endothelium contains one layer of squamous or elongated endothelial cells located on the basal lamina. The subendothelial layer is located below, and in larger elastic arteries, this part is composed of connective tissue with collagen and elastic fibers. Smooth muscle cells are also present in the subendothelial layer. The internal elastic membrane is located between tunica intima and tunica media, but it is not well distinguished because It is one of many elastic layers which are present in the wall of the elastic artery. The internal elastic membrane is usually identified only because it is the innermost elastic layer of the arterial wall. The tunica media of elastic arteries is the thickest part in the three layers of elastic arteries. This part is composed of fenestrated lamellae that are arranged in concentric layers. The number and thickness of these lamellae are related to blood pressure and age. The smooth muscle cells are spindle-shaped with an elongated nucleus, and they are less abundant. External elastic lamina is located between tunica media and tunica adventitia. The layers that make up the wall of the aorta are shown in this slide. This is longitudinal section of the aorta, but two layers are visible only: tunica intima and tunica media. The adventitia is not visible in the specimen. Tunica intima consists of a lining of endothelial cells with basal lamina. The endothelium resting on the subendothelial layer with connective tissue. This part contains collagenous and elastic fibers. Smooth muscle cells are also present in the tunica intima of the aorta. The endothelium is difficult to preserve in the aorta and is frequently lost. Very thick tunica media contains large amount of elastic material. The elastic material appears as elastic fibers and as fenestrated membranes. This specimen is stained to demonstrate elastic fibers. They look like dark, wavy lamellae. The smooth muscle cells are not visible in this staining. The internal elastic membrane is not well distinguished because tunica media contains numerous elastic fibers. The tunica adventitia in the aorta is relatively thin. This part is composed of connective tissue with collagen and elastic fibers. The elastic fibers don't form lamellae than those in the tunica media. The principal cells are fibroblasts and macrophages. There are vasa vasorum and nervi vascularis in this layer. Small blood vessels provide nutrients through the outer portion of the thick arterial wall. The inner part of the thick wall is supplied by nutrients from the blood in the lumen of the vessel. This slide demonstrates microscopic specimen. This is a longitudinal section of the aorta. We used here special staining for showing elastic fibers. we can see that elastic fibers are numerous in the thick tunica media. The outermost layer, this is adventitia, light pink in our picture. This part is usually less than half the thickness of the tunica media. Tunica intima in this specimen is not well visible. Elastic arteries branch and become medium-sized muscularis arteries that are the most numerous vessels in our body. Muscular arteries are also called medium arteries or distributing arteries. Their tunica media contains more smooth muscle and less elastic fibers than do elastic arteries. They distribute blood to various organs. We can see here medium or muscular artery and medium vein. The artery is located left, the vein is located right. The size of medium arteries depending on the location. The wall is dominated by smooth muscle. We can see here elongated purple nuclei of smooth muscle cells. There are 6 to 40 layers of smooth muscle cells arranged in a circular orientation around the lumen. When smooth muscle contracts, the size of lumen decreases and blood pressure increases. Please pay attention to the thickness of the artery and vein wall. Tunica intima in the muscular arteries is thinner than that in the elastic arteries. It contains endothelium and subendothelial layer that contains a few smooth muscle cells, also in contrast with that of elastic arteries. The internal elastic lamina or internal elastic membrane located between tunica intima and tunica media is prominent and forming the boundary between these two layers. The internal elastic lamina is very well distinguished and very well visible when we use special staining for showing elastic material. This is muscular artery. We use here special staining for elastic to showing the localization of elastic fibers. Elastic tissue is largely concentrated as two well-distinguished sheets. Internal elastic membrane and external elastic membrane. The subendothelial layer is sometimes so scanty that the basal lamina of the endothelium appears to make contact with the internal elastic membrane. In histologic slides, the internal elastic membrane appears as a well-defined undulating or wavy structure because of contraction of the smooth muscle. The tunica media contains little elastic fibers because the major component of this layer are smooth muscle cells which are not visible in this staining. On the periphery of the tunica media, external elastic membrane is present. It is less prominent and more variable. The tunica adventitia consists of elastic fibers that are visible in our picture. Collagen fibers and ground substance are also present in this layer. Again, muscular artery. Note the tunica intima. The tunica intima is usually a very thin layer, not visible at low magnification. Thick tunica media contains smooth muscle cells. We can see here elongated nuclei of smooth muscle cells. The tunica media of muscular artery is composed of concentrically located smooth muscle cells with scanty elastic fibers located between them. Larger muscular arteries may have as many as 40 layers of circularly arranged smooth muscle cells. This is a light micrograph of muscular artery. The subendothelial layer is scanty, that the basal lamina of the endothelium appears to make a contact with the internal elastic membrane. Internal elastic membrane appears as well-defined wavy structure. Tunica media, thick tunica media consists mainly of circularly arranged smooth muscle cells, elastic and collagen fibers. We can see here elongated nuclei of smooth muscle cells. Tunica adventitia consists mainly of connective tissue. We have here very well defined three layers: tunica intima, tunica media, and tunica adventitia. This slide shows muscular artery located right and vein located left. The artery is well defined with thick wall and smaller but regular or open lumen. The vein is poorly defined with a large irregular lumen. The wall is definitely thinner than the wall of the artery. The tunica adventitia of the muscularis arteries consist of elastic and collagen fibers and ground substance with dermatan sulfate and heparan sulfate. This extracellular matrix is produced by fibroblasts in the adventitia. Vasa vasorum may also be present. Muscular artery. The wall is divided into tunica intima, tunica media, and tunica adventitia. The internal elastic membrane and external elastic membrane are also visible. This is schematic diagram that demonstrates the histological structure of the elastic and muscular artery. The tunica intima of both vessels contains endothelium and subendothelial layer that is thinner in the muscular artery. The tunica media is the thickest layer in the wall of elastic and muscular arteries, but the main component of the tunica media of elastic artery is elastin, with numerous elastic fibers that form fenestrated membranes. The main component of the tunica media in muscular artery are smooth muscle cells, which are arranged in 40 layers. The tunica adventitia of elastic artery contains more elastic fibers. Both contain connective tissue. Small arteries and arterioles are distinguished from one another by the number of smooth muscle cell layers in the tunica media. Arterioles are the smallest arteries, leading blood flow into capillaries network. Tunica intima consists of endothelium and thin subendothelial connective tissue with collagen fibers type 3 and a few elastic fibers. Both internal elastic membrane and subendothelial layer are often absent. A thin fenestrated internal elastic lamina may be present in larger arterioles. The tunica media is composed of one or two layers of smooth muscle cells that completely encircles the lumen. They have a very small diameter and narrow lumen. The tunica adventitia contains loose connective tissue, and this part is limited or absent. We have here schematic diagram of elastic artery, muscular artery, and arteriole. The wall of all blood vessels which are present in our picture is divided into three layers: tunica intima, tunica media, and tunica adventitia. Tunica media is the best developed and is the thickest in the elastic artery. It is composed of elastic fibers which form fenestrated membranes. Tunica media of muscular artery is a little thinner than the tunica media of elastic artery, and it is composed of smooth muscle cells. Muscular artery has a little thicker tunica adventitia. The tunica media of arteriole contains one or two layers of smooth muscle cells, and the tunica adventitia is very, very thin or absent. The arterioles branched into small vessels which are surrounded by a discontinuous layer of smooth muscle called medarterioles. Medarterioles branched into capillaries. The wall contains tunica intima with endothelium and basal lamina without subendothelial layer. Tunica media is composed of incomplete single layer of smooth muscle cells, and the outermost layer, tunica adventitia, contains sparse loose connective tissue. Traditionally, veins are divided into following types on the basis of size: large veins, small and medium veins, and venules. This slide shows the vein. The black arrows show the large irregular lumen of the vein. The wall is thinner than the wall of the artery. The following layers, tunica intima, tunica media, and tunica adventitia, are difficult to distinguish. We don't see the boundaries between this layer. Tunica adventitia is about twice as thick as the tunica media, and this part is the best developed in the vein. Veins with a diameter greater than 10 millimeters are classified as large veins. The tunica intima is composed of endothelial cells with basal lamina. Below thick subendothelial layer is located. Very often the boundary between the tunica intima and tunica media is not clear. Tunica intima may form valves in some veins. The tunica media of large veins is relatively thin, and this part is composed of circumferentially arranged smooth muscle cells, collagen fibers, elastic fibers, and some fibroblasts. The tunica adventitia of veins is the thickest layer of the vessel wall. This part is composed of collagen and elastic fibers. Numerous fibroblasts are also present in this part. Tunica adventitia also contains longitudinally disposed smooth muscle cells. The tunica intima of medium and small veins contains endothelial cells resting on the basal lamina. Below, thin subendothelial layer is located. Subendothelial layer contains smooth muscle cells scattered between the connective tissue elements. The tunica media of medium veins is thinner than the same layer in medium-sized arteries. This part contains smooth muscle cells circularly arranged, collagen fibers, and elastic fibers. This part may also contain smooth muscle cells longitudinally arranged, which are located beneath the tunica adventitia. The tunica adventitia is thicker than the tunica media and contains collagen fibers, network of elastic fibers, fibroblasts, and few scattered smooth muscle cells. Venules receive blood from capillaries and have a diameter as small as 0.1 millimeters. Most venules are muscular, with at least a few smooth muscle cells in the wall. Tunica intima of venules is composed of endothelial cells with basal lamina. Very thin tunica media contains sparse connective tissue with few smooth muscle cells. Tunica adventitia, the outermost layer, is also very thin, contains some collagen and few fibroblasts. Valves are the characteristic histological feature of the veins, and valves are absent in the arteries. Each valve is composed of delicate semilunar projections of the tunica intima of the vein wall. The projections consist of fibroelastic connective tissue, and they are lined on both sides by endothelium. Each valve consists of two leaflets, the free edges of which project in the direction of blood flow. Valves are present in veins of more than 2 millimeters in diameter, particularly those draining the extremities. We have here two microscopic slides stained with chematoxylin and eosin, two veins with valves. And we can see here that the valves are folds of tunica intima, and because of that each valve is covered by endothelium. The valves are composed of connective tissue. We have here characteristic histological features of vein and arteries, and we start from the lumen. Artery has regular open lumen. The histological features of the arteries are located left, vein right. The lumen of vein is irregular but larger. The lumen of artery is usually smaller. Three layers are very well distinguished in the wall of artery. The borders between three layers are not clearly visible in the wall of vein. The best developed layer is tunica media in the artery. The best developed layer in the vein is tunica adventitia. Valves are absent in the lumen of arteries. Valves are present in the lumen of veins. The wall of arteries is thick. The wall of veins is thinner. In the upper part of our slide, we have schematic diagram of artery and vein, and below we have microscopic slide, artery and vein. And please look, artery, small regular lumen, vein, lumen irregular but larger. The three layers, tunica intima, tunica media, and tunica adventitia, are very well distinguished in the artery. We can see very well the boundaries between the layers. The three layers are not well distinguished in the vein. The best developed is tunica media in the artery. Tunica adventitia is the best developed in the vein. The wall of artery is thick, the wall of vein is thinner, and the veins may contain valve. Valve is absent in our microscopic slide, but the arteries don't contain valve in the lumen. Very similar picture what previously. We have left artery with tunica intima, tunica media, and tunica adventitia, and we have here, I think, muscular artery, because we have a lot of smooth muscle cells, and right vein is located, and in the middle we have microscopic slide of artery and the vein. This is microscopic slide stained with chematoxylin and eosin. We can see artery left and vein right. And please look at the lumen of artery small but open and regular, lumen of vein irregular but larger. The three layers are very well distinguished. The thickest is tunica media in the artery. The layers in the wall of vein are not well distinguished, and the best developed, the thickest, is tunica adventitia. This vein doesn't contain the valve in the lumen. And last one picture for today, schematic diagram of the structure, the wall of artery left and the wall of vein right, and we have here tunica intima, tunica media, and tunica adventitia. And in this picture, tunica intima forms valves which are present in the lumen of the vein. Thank you for your attention.